Hello students. Today we will discuss about Peziza. Peziza जो है it is a saprophytic, very often coprophilous in habit. It frequently grows on dung, rotting wood or rich humus of forest soil. Some species of this genus grow on burnt or charred wood. The species commonly found is Peziza vesiculosa. It is one of the larger cup fungi and is cosmopolitan in distribution. Now we talk about its mycelium. Mycelium it is well developed, frequently perennial, and consists of a dense network of hypha. The hyphae are branched and septate. The cells are uninucleate. The hyphae are hidden from view as they ramify within the substratum. They form a complex system which extracts nourishment from the substratum. The fruiting bodies are above the ground. Asexual production. It takes place by the formation of conidia and chlamydospores. The conidia are exogenously formed spores. They are abstracted from the tips of conidiophores. Each conidium germinates to form a new mycelium. The chlamydospores are thick-walled resting cells. They are intercalary in position. They may be formed singly or in series within the cells of hyphae. Under suitable conditions, each chlamydospore germinates and gives rise to a new mycelium. Sexual reproduction. The sexual apparatus is wholly lacking in Peziza vesiculosa. This does not prevent the development of a fructification or the apothecium. These are the apothecia. Apothecium, it is aerial and relatively a short-lived structure. The sexual process thus takes place. It is extremely simplified and consists of the association of two purely vegetative nuclei in a pair. The adult mycelium consists of a tangled mass of hyphae. Certain vegetative cells in the center of the tangled hyphal mass have been seen to possess nuclei which becomes associated in pairs. These pairs of nuclei are called dicaryons. The dikaryotic condition is brought about either by autogamous pairing or by somatogamous copulation between the vegetative cells or the adjacent hyphae of tangled hyphal mass. The cells with the dicaryons give rise to exogenous hyphae, which becomes multicellular by cross walls. Their cells are binucleate. The terminal binucleate cell of each exogenous hypha functions as an ascus mother cell. Formation of croziers in the development of asci has not been reported in Peziza vesiculosa. The exogenous hyphae and the dikaryotic cells from which they are developed together with the ascus mother cells represent the dikaryophase in the life cycle of Peziza. The two nuclei of the ascus mother cell fuse to form the synkaryon. The young ascus with the synkaryon represents the transitory diplophase. The synkaryon undergoes three successive divisions. Of these, the first and the second constitute meiosis. This results in the formation of 8 haploid nuclei which become organized into ascospores. The mature ascus is an elongated cylindrical cell, as you can see in this diagram. The ascus wall is lined by a thin layer of cytoplasm or epiplasm, which encloses a central vacuole filled with sap. In the vacuole lie the oval ascospores. The erect asci lie side by side, lining the cavity of cup-shaped apothecium. You can see. The sky near the margin of the cup bent towards the source of light, being positively phototropic. Interspersed between the sky of the sterile hypha are called paraphyses. These are the paraphyses. The rest of the apothecium consists of densely interwoven branched hyphae forming a pseudo-parenchymatous tissue which supports the hymenium. The apothecia are sessile or shortly stalked cup-shaped structures, regular in form and large in size, varying from 2 cm to several inches in diameter. In Peziza vesiculosa, the apothecium is of pale fawn color, but Peziza aurantia has brilliant orange apothecium. Now, if we talk about the salient features of Peziza, Peziza is a saprophytic discomycete which is often coprophilous in habit. A species growing on rotting wood and rich humus are also plentiful. The mycelium is inconspicuous as the branched septate hypha constituting it ramify within the substratum. The cells are uninucleate. Asexual reproduction takes place by means of non-motile spores of two kinds, namely conidia and chlamydospores. The sexual apparatus is lacking in Peziza vesiculosa. The sexual process is extremely simplified. It consists in the association of two vegetative nuclei from a dikaryon by the somatogamous copulation of adjacent hyphae or by autogamous pairing. Ascogenous hyphae arises from the dikaryotic cells. They become septate and branched. The cells are binucleate. The terminal binucleate cells of the ascogenous hypha or their branches function directly as ascus mother cells. Formation of croziers has not been reported in Peziza vesiculosa. The ascogenous hypha, the dikaryotic cells from which they are developed, and the binucleate ascus mother cells constitute the dikaryophase in the life cycle. It is parasitic. The two nuclei in the ascus mother cell fuse to form the synkaryon. The young ascus with a synkaryon represents the transitory diplophase. The ascus cell advances towards maturity. The synkaryon undergoes 3 successive divisions. The first 2 constitute meiosis. The resultant haploid nuclei are organized into 8 ascospores in the mature ascus. The ascocarps in Peziza are large, fleshy, red to orange, cup-shaped epigean, sessile or subsessile apothecia. So this was all about the Peziza. This is the taxonomic position. If we talk the classification of Alexopoulos, he divided Peziza into division Mycota, subdivision Eumycotina, class Ascomacetes, subclass Euascomatidae, subseries Hymenoascomacetes, subseries is Discomacetes, order is Pezizales, family Pezizaceae, and the genus Peziza, and the common species is Vesiculosa. And if we talk about the classification of Alexopoulos and Mims, in 1979, they divided Peziza into superkingdom Eukaryota, then Mycete kingdom, A Mastigomycota, Ascomycotina, class Ascomacetes, subclass Hymenoascomacetes, series Discomacetes, order Pezizales, family Pezizaceae, genus Peziza, and the species is Vesiculosa.